Thank you very much for, for the opportunity to talk here. And I'm very honored to give a talk after Professor Chen. And so, yeah, I am very much inspired by using OPTIR to look on amyloids and to look on amyloids in tissue. So that's First of all, I would like to thank my students and collaborators, and of course, Photothermal and Grant Agency for the work that I am doing. I never have time to thank them, as I'm usually speaking slowly and go over my time. So I do it now. So my group has 3 instruments. So we use FTIR, we use OPTIR, and we use nano-IR. And the story began in 2017 when we looked on primary neurons that overexpress amyloid proteins. And for the first time ever, we saw that protein not only aggregate but also accumulates inside of neurons. However, the resolution was absolutely not enough to further dig down into this phenomena. So after that, I was super lucky to learn OPTIR technique and been in synchrotron Soleil at 2018 to use for the first time OPTIR simultaneously with synchrotron-based FTIR on the brain tissue slices. So, and I really value this technique because this is non-contact technique. It's not destructive, and it gives submicron resolution, which is extremely important when we talking about multimodal sample characterization. For example, after OPTIR, after scanning tissue with OPTIR, it is possible to do other spatial techniques like proteomics or transcriptomics. And that gives not only spectra, it also helps to interpret the spectra and understand biological phenomena in a more detailed way. So here I show a brain tissue, and white spots show that it's amyloid plaques in mouse model of Alzheimer's disease. And the red image close to that one show how we see those proteins using immunofluorescence. So we specifically see presence of amyloid protein in the tissue. However, we can see nothing about amyloid structures. And then the next image shows actually structure of this is amyloid plaque in OPTI at the in infrared image. So, and if we take spectra, we could clearly differentiate polymorph heterogeneity that present in the core and in corona and pericore plaque and pericore area. So even more, we could apply same technique and look on amyloid plaques in living tissue. And that was very important project that we did during pandemic and published in 2023. And since then, we're working more on that in that direction. So far we showed that it's not only brain tissue that can be possible to examine, but also other ex vivo fresh biopsies just taken from the tissue and deposited on a slide can provide good spectra. So we worked further, and this is unpublished data that the paper is in revision right now. So where we look on organoids, and here we show that those organoids can be characterized with OPTIR, and this is very important because, as you could see, organoids, it's a mini organ, and when during differentiation, there is a spatial distribution of cells, and those cells, they get different access to the media, and therefore they can develop in different ways. And using OPTIR, we looked on those organoids and assessed cellular heterogeneity in those organoids to make sure to make sure that all cells converted from skin cells to neurons. So, and actually OPTR help us to find the right window for the tissue, for the cell incubation. And we actually shortened the protocol for organoid conversion. Also, we looked on the cells, and in those cells, we looked on different amyloids, specifically on tau and alpha-synuclein, and we described a lot of those amyloids that can simultaneously present in the tissue, and discuss their structure that were detected directly in tissue. So this paper is just released, and so if you would like, you could look at that. So, however, I still think that the main one of the important achievement was that we could look on amyloids directly in cells and at very high resolution. Here you could see neuronal spine. Neuronal spine is a tiny protrusion, usually about 1 or 2 micron mushroom organelle sitting on the top of neuron, and actually their memory is stored, and this is the organelle that involved in memory storage and retrieving. And the question was, do we have formation of amyloids in those organelles or not? And using OPTR we actually were able to answer this question. So, and the reply is a little bit counterintuitive. So we have accumulations but not aggregations of amyloid in those in those compartments. So by that, I would like to say that OPTR is very sensitive, and spatial resolution is perfectly matching immunofluorescence imaging, which is a core technique for biology. So together with Professor Chen, we have incredibly interesting project. We look on the tissue, and we use multimodal approach. So using nanotomography, we look for area of interest in spinal cord, and then when we find our area of interest, we slice the tissue, and we start tissue analysis. So with infrared spectroscopy, we look on distribution of amyloid plaque, and we look on distribution of amyloid plaques in a really big area in all sections. So we in that way we plan to reconstruct 3D 3D volume, and also those sections are going to be immunostained, and we will get another 3D volume that characterize amyloid plaque distribution in tissue. And here, and question here is extremely important. So we want to really see that observed plaque heterogeneity that was documented previously, is it a true event or due to postmortem processing when the different plaques hits at a different level? And sometimes we have core and sometimes we have corona. So in 2D, we clearly differentiate those two types of amyloids. However, it is not clear if they are really exist. So with this project, we're planning to answer this question. So here is how it's working. So we take tissue, we do nano-CT, we find this is how look like amyloid plaque in tomography. So then we label, then we doing spectroscopy, and then we label these amyloids. And of course, we label not only for amyloids, we label for other cells that present in the tissue to link those structure with those functions. So yeah, this is the project, and with that we're planning to link amyloid structural variants to clinical phenotypes. So I hope that I showed you that OPTR is excellent tool to look on amyloid differentiation in cells, in extracellular tissue, and also is useful for organotypic cultures differentiation. And taken together with other technique, we can really link amyloid structures to Alzheimer's disease clinical phenotypes. So with that, I really thank you all for attention, and if you have questions, I'm happy to take.
Thank you, Oksana, for sticking the time, which means we do have time for a question. And so, question that you've got is
That's the wish. We physically cut them.
Well, thank you. I'll move on to Mark, but I just want to say, I think, Oksana, you're particularly special for me in that you're an actual biologist who's using spectroscopic tools. And for that, I think, I place a special weight on your research because you understand the problems and how we can help solve them. So thank you
so much.